Hi, we know that genomes, one of the essential properties of all genomes is to undergo replication. And we also know that there are errors that happens because of misreading, because of tautomerism, many other factors. There are going to be mistakes done by DNA polymerase. Of course, DNA polymerase is a very efficient one in the sense that it has to polymerize a lot of bases in a given span of time. So mistakes are inevitable. In that sense, if we can expect that replication is essential, and then because of replication there are going to be errors, then we should be expecting that generation after generation there is huge variation, and that is mutations. And because of those mutations, or because majority of the mutations are detrimental, we should expect the extinction of that particular genome in a while. So there are mechanisms to undo the mistakes, and that is what we will discuss now. I am Dr. B.C. Amar, Assistant Professor at Shastra. The one dedicated repair mechanism to scan through all the errors that the replication does, that happen during replication, especially the mispairing, that is incorporation of a wrong base, is called as mismatch repair. And there are some essential proteins that are involved in it, which are called as mutase. And then there are many other mutations that are, there are many other proteins that are involved in, that are involved in this mechanism of repair. So here there is a mismatch, that is T base paired with G. And then one of it is a template, which we will discuss in detail in a while. And the other strand is the primer, or the growing new strand. So mutase proteins, as a timer, it keeps on following the replisome. As the DNA polymerase is synthesizing, mutase keeps on scanning right behind them. At least in most of the cases, that is how it can. There is a way to figure out the mismatches that happen during replication and then eliminate those. So mutase dimer keeps scanning the genomes for mismatches, and once it identifies a mismatch can be easily identified because the DNA is kind of a little bit distorted at that location because of the mispairing. And mutase changes, it stalls at the distortion or the mismatch, and then it will bind to it. There are conformational changes because of which it will bind to ATP, and using that ATP there are furthermore conformational changes in this region, and that will allow or increase the affinity for MutL, another protein. And there is MutL, the job of it is to recruit another protein, that is MutH. The MutH is an important enzyme, and the MutH is the one that cleaves the one of the strands, and then there will be recruitment of UARD, which is an helicase, which will remove one of the strands. This strand is eliminated now. And now there is an opportunity for DNA polymerase to take up this sequence and synthesize this strand again, and there is a new opportunity for adenine to be added at the opposite to thymine. Yes, let's quickly again look at it before we go further. Mutase is, the job of it is to detect the distortions, and once distortions are bound, there are conformational changes because of which ATP is bound. There will be further conformational changes, and then there is recruitment of MutL. The function of MutL is to bring MutH. MutH is the one that nicks the DNA, that is, makes a single break on the backbone. And then we have the helicase activity of UARD, which will remove the strand from here until here. And then DNA polymerase 3 will fill up the gap, and ligase will seal the gap. And now we have the mismatch has been repaired. The story is not yet over there. The question arises, is, say we have a TG, and which one do we think is the original one? Which one should be replaced and which one should be retained? Should T be repaired or removed, or G be repaired? That is, if we follow there are simple mechanisms of how the repair mechanisms can distinguish and remove only the newly added one. The parent strand is retained, parent nucleotide is retained, whereas the other strand is removed. Say for example, we will go slowly with this. There are GATC sequences. We always read from 5' to 3'. So GATC sequences like dimethylase. Dimethylase is an enzyme that recognizes specifically the GATC sequences, and it methylates the adenine. At carbon 6 there is an amino group, and that amino group it adds a methyl group. So the template, the DNA, parent DNA strand will have methylation on both the cases. I hope you can recollect that the same mechanism is used for regulation of replication initiation. There we have seen about SECase ability to bind to the hemimethylated DNA. Very similar mechanism also happens here. So we have GATC sequences and the adenines are methylated. If replication was happening, replication would allow a new strand synthesized, but this adenine is not yet methylated. And so as a result, what we will have, these kinds of sequences are referred to as hemimethylated, because one side of one strand is of the GATCs of one strand are methylated, whereas GATCs of the complementary strand are not yet methylated. So that will allow the protein MutH. MutH always binds to hemimethylated region, and again we should remember that MutH does not bind to every hemimethylated DNA. It needs MutL to be there, and for MutL to be in that location, they need MutS in a proper conformation, and that is how MutS is in the mispaired bases, and MutL is recruited, and that allows the recruitment of MutH. And MutH binds, and it has an endonuclease activity. It binds to hemimethylated DNA, and it cleaves only the strand that is unmethylated. That is how parent strand and the newly formed strand can be distinguished. It is always the newly formed strand that is repaired. There is yet another problem, of course, is, say for example, if we have there is a possibility that GATC is about 4 bases, the probability is a little high. Nevertheless, there is a chance, as you can see here, there are 2 scenarios. In one scenario we have, this is the distortion, and in this case here is the distortion. So in one side we have methyl group on this side. There is also a possibility that sometimes we may have methyl group on the 5' side, like as shown here. So we need an exonuclease activity. The same proteins are not useful in both the cases. The recruitment of MutS, distortion is important for the recruitment of MutS or conformational changes, then recruitment of MutL, and then recruitment of MutH. It is same on both the cases. Here is MutH, and this is MutL, and this is MutS. So using nickase activity, nick has been made on both the sides. In one case, the nick is on the 5' side of the distortion here, and in another case, the nick is on the 3' side in relation to the distortion or the mismatch. So we need different exonuclease activities. In sense, we have in this case the mutation should be, the exonuclease activity should be in this direction to remove this the misincorporated base, and in this scenario we need in this way, in this direction. So we have 2 different exonucleases. One is called as EXO7, which has 5' to 3' exonuclease activity, and there is another one, EXO1, which has 3' to 5' exonuclease activity. So they will fill up, they will empty the space, which can be taken up by DNA polymerase, and they can fill up the gaps in this direction, and then ligase will seal it up. That is how the mismatches can be removed. So quickly to summarize, we will see there is mutase, there is mutase will detect the mutation or the mismatch, then we have mutal and mutage, and then we have the helicase activity, the exonuclease activity as well. In the exonuclease activity, we have two different proteins based on which side it is, which side the nick has been made. The nick is made by mutage based on hemimethyl status on the strand that is unmethylated, and we have EXO7 with 5' to 3' exonuclease activity, and EXO1 with 3' to 5' exonuclease activity. This is how a majority of the mismatches are eliminated right after the replication. I hope that is clear. Thank you very much. See you again.